Hi, everyone. Thank you for joining us for our very first webinar. We have registrants from all over the world. I just wanted to kind of read off a few of the countries we have represented here. We have the United States, Canada, Italy, Sweden, UK, France, China, Japan, South Korea, Ireland, and South Africa. So we definitely have a very international audience today, which is really great. My name is Monica Engebretson. I'm the North America Campaign Manager and Head of Public Affairs for the North America campaigns for Cruelty Free International. And just some housekeeping stuff. If you've not been on GoToWebinar before, there's a questions bar on the side. If you have questions throughout the webinar, you can go ahead and type those in. We can either email you privately to answer those, and we will try to have some time at the end of the webinar to go through questions. But as you have those questions, go ahead and put them in there, whether they're for me or for Carol. And everybody should be able to see my screen, and I'm just going to jump right in and introduce our first speaker, who will be walking you through a basic overview of non-animal tests for assuring cosmetic safety. Dr. Carol Treasure is the founder and CEO of Accelerate, an animal-free testing laboratory. Accelerate conducts regulatory and in vitro safety and efficacy tests for global cosmetic ingredient manufacturers as well as high street retailers. Carol founded Accelerate in 2008 with a mission to accelerate the world's transition to 100% animal-free testing, and the company's work has been recognized at the regulatory level and by the OECD and by top funding bodies such as Innovate UK and Horizon 2020. And with that, I'm going to switch the presentation over to Dr. Carol Treasure, I think. And yes, I want to make her the presenter. So it should be switching over, and I'm going to go ahead and turn my camera off for now. And I can see your screen, Carol.
Great. Okay. Thank you very much, Monica, for the introduction, and welcome everybody, whatever time zone you're in. It's great to have you with us today. And what I'm going to do is just talk you through some of the key elements of how we are able to develop safer cosmetics through animal-free testing methods. I'm just waiting for the slide to move on.
You may need to click onto your presentation and then push the arrow key on your keyboard, maybe to get it to advance.
I'm actually trying to escape out of this mode so I can move the slide on, but it seems to have frozen up, unfortunately. It didn't do this in the test run, did it, Monica?
No.
My screen seems to be frozen, so oh, can you see
there we go.
Slide now?
Yep, there you go.
Well, let's hope the next one comes up okay. I just wanted to start the presentation thinking about why this is an interesting topic to us. And at the end of the day, this is all about improving human safety, but without making compromises on either animal welfare or on the environmental impact of what we're doing. So I'd like to just give an introduction to animal-free testing to start with. And I know we have a variety of attendees in the audience today, and some of you are very familiar with the topic, but many are not. So just bear with me, I'm going to give some basic definitions for those not directly involved in the field. So just to clarify, an in vivo process is something that's performed using a whole animal. It also refers to studies in humans. An in vitro process is performed outside of the living organism. So that can be an experiment that's conducted in a test tube or a culture dish. So it's essentially the opposite of in vivo. And then we have in silico, we'll be mentioning that today as well. and that's a process performed on a computer or via computer simulation. And in general, when we mention animal-free testing or non-animal methods, we're referring to a combination of both in vitro and in silico. I'd like to start by just thinking about the journey that technologies have taken over the last few decades. You may be familiar with the concept of the 3 R's, which is the reduction, refinement and replacement of animal testing. At Accelerate, we're really focused on the 1 R, which is replacement, and that's the topic that we're talking about today. And it was only really a few decades ago that all cosmetic testing was taking place in animal in vivo models. And then through the 1990s, which is when I did my PhD at the Frame Lab here in the UK, experiments were really starting to gain momentum using cultured human cells. And at the time, those tests were really heavily criticised as not being adequate to model human physiology. And I have to agree, I think that we all knew at the time that those methods were very simple, but we had to take that first step. And if those labs working in the area at the time hadn't have developed those cell culture models, we wouldn't have had that explosion of technology, which we see in the main part of this slide. And just thinking about all of the technologies that are available today, to give us much more advanced in vitro systems with maximum human relevance. It's really inspiring to me to think about how far we've come over the last few decades. And I think certainly if I had known as a young student that these would be the kind of tools that we had access to, I certainly would have been really excited by that at the time. So you can see in the circle here, we've got a number of exciting in vitro technologies, things like tissue engineered human skin, human airway epithelium, and we now have systems, you may have heard of organ on a chip, which is essentially a series of very miniature organs which are linked up with an artificial circulation so that we can study on a tiny scale how organs of the human body interact with one another. We also have access to genomic screens, which use sophisticated technology to look at how genes are regulated in response to chemicals, a whole range of mechanistic tests and culture systems, even down to 3D bioprinting of models. So I think what I like to take from this slide is that inspiration of where we've come scientifically, and really in a relatively short period of time, we now have this toolbox available. And if we look back to the beginning, to using animal models, you can see easily how out of date that now seems. It's a very powerful approach. And what we need now really is for governments, regulators and industry to be taking up these methods, and I hope to demonstrate today that that is very much the case, to obtain international harmonization on this progress. Finally, just before I move on from this slide, I just had a little fact in the left-hand side there that states that more than 90% of drugs which are tested on animals in the pharmaceutical industry fail in subsequent human trials. We've always known that animal tests are flawed models, and now that we have these amazing tools available to us, I think that's all the more reason to shift to them, whether that's for cosmetics or for drugs or for any other class of chemical. So given all of this progress that we've seen, I always like to question whether alternatives are actually still the alternative. I try to avoid using that word because alternative somehow seems to imply second best, and the technology that we're looking at certainly isn't that way. We're looking at scientific advancement. And what we see now is that some of the alternatives are now being adopted as the mainstream and as the default method for regulatory safety testing around the world. And we've seen with these methods extensive validation trials, which have demonstrated enhanced human safety. We have a large amount of high quality data now that's been gathered over a number of years, and a very high level of confidence in these methods. So perhaps the alternatives are now becoming the mainstream. When we think about how these tests are actually applied in real life, I think that a good starting point for us to consider is the OECD Test Guidelines Programme. The Organisation for Economic Cooperation and Development now has a wide variety of non-animal tests available with full international regulatory approval for use in any of the OECD member countries. And it's worth noting that the OECD operates on the basis of mutual acceptance of data. And what that means is that test results that have been generated in a GLP accredited lab in one member country can be used in any of the others. And that means that we have a lot of harmonization, and it brings economic, ethical and environmental benefits, knowing that the tests don't have to be repeated again and again in different countries. So what the OECD Test Guidelines Programme does essentially is that provides us with a toolbox to enable us to comply with regulations that are coming in around the world, including bans on animal testing for cosmetics. They are the means to the end, really, because they're the methods that we can put our faith and confidence in to make a safety assessment. Within the OECD guidelines, the human health endpoints are covered in section 4. And you'll see throughout the presentation that I've added a number of live links, which you're very welcome to follow for further resources when you receive the recording of the webinar. We think about the key tests that are required and the ones that have been addressed by the OECD. There's a lot of commonality with most regulatory frameworks around the world requiring the assessment of a well-defined set of human health endpoints. And the key ones for cosmetic ingredients are skin irritation and corrosion and sensitization. And then we've got eye irritation, mutagenicity and acute toxicity. Now, I should point out that these are the key human health endpoints that are required in the EU REACH regulation. So I'm using that as an example a little bit today because it's one of the most established regulations around the world in terms of safety assessment for chemicals. And these are the key endpoints that are required for any chemicals being imported into the European Union in 1 to 10 ton annual quantities. If we go up to higher quantities, there are a few more endpoints, and I'll discuss that a little bit later. So what tests do we have available for these human health endpoints that need to be demonstrated in terms of non-animal testing? Well, it's a very positive picture, and actually that's what I wanted to be the takeaway message from this initial slide. Again, I've provided live links there to each of the tests, so feel free to check those out at a later date. But you can see that for skin corrosion, irritation and eye irritation, we've got a number of tests listed here, which actually make use of some of the technologies that I showed on the introductory slide. So the reconstructed human epidermis and the reconstructed cornea epithelium are tissue engineering technologies to create human tissues in the laboratory. And there are others that are using artificial membrane barriers, human cell lines, for example. But there are a couple of tests on here which I should point out, even though they are they're formally considered as animal-free tests, but they're not technically animal-free, and that's where some caution is required in the interpretation of these lists. So we have Test Guideline 430, the transcutaneous electrical resistance method for skin corrosion. For example, that actually requires animal sacrifice to provide the skin that is used in the test. And in some countries, that's considered a live in vivo animal experiment because of the treatment it is required prior to sacrificing the rats for that study. And we also have the bovine corneal test, which is the BCOP test. That's an eye irritation test that uses a bovine cornea obtained from slaughterhouses. So it's beyond the scope of this talk today, really, but we're very keen at Accelerate on demonstrating that not all in vitro tests are equal. Some of them do still use animal components. This is just to round off and complete the list. So for now, I'd like you to just kind of get the takeaway message that actually there are a lot of in vitro tests available for the key human health endpoints. And these are the ones for skin sensitization and genotoxicity. So having shown a list and demonstrated that there are a lot of tests available, what I'd like to do is to just give an overview of the technologies and to just, without going into too much technical detail, which isn't really the point of today's webinar, but just to give an overview of the type of tests that are being used at OECD level. Firstly, if we take a look at the skin irritation and corrosion testing, the picture that you see on the right here in the circle is a cross-section through a tissue-engineered piece of human skin. And this is the same technology that's used to develop skin grafts for burns patients. And that same technology has been developed in parallel over the years to provide tiny little discs of skin, which you can see in the dish in the photos at the bottom of the slide. And these are a very, very good representation of human skin in terms of the morphology, the structure and the function. And the pink stained part that you can see at the top there, that's the human skin barrier. So these models actually are able to simulate absorption of cosmetic ingredients and products across the skin. These models are suitable for testing ingredients or finished products. They're a great model of real-life exposure. And similar models are used for skin irritation and corrosion testing. And what happens is those ingredients are applied to the skin surface. They're incubated for a set period of time. And then we're able to measure the damage to the cells over that time. And using a validated prediction model, to then make a prediction whether that ingredient is going to be a skin irritant or corrosive. Now, there's a really long history of safe use of these test guidelines. The one for irritation has been in place since 2010 and the guideline for corrosion since 2004. We move on to think about eye irritation testing. This is another very common test for cosmetic ingredients and products. If we start our kind of tour of this slide on the left-hand side, and you can see that here we have a cross-section through the bovine cornea. So this is the test that I mentioned just now. It's not totally animal-free, but it's conducted using the cornea obtained from the slaughterhouse. If you look here at the picture, you can see a cross-section and the structure of the cornea. That cornea is taken and placed into a chamber. And then the researchers will then measure the opacity, so the effect on the cornea and how cloudy it becomes and how it increases its permeability to chemicals over time. Now, I think that one of the striking things for me when looking at these models together is to now take a look at the human corneal model, because we've demonstrated here again through tissue engineering technology that we're able to construct in the lab a really good simulation of the cornea. And moreover, this is using human cells, so we have a more direct relationship between this model and genuine human physiology, in addition to knowing that these models are freely available and they're very reproducible. So they have other advantages, as well as the fact that they're using human tissue. There are some limitations of using the human corneal model in terms of classifying an eye irritant according to some of the subcategories that are required for a full safety assessment. And just with any of the animal models that we have available, there are also some drawbacks to the in vitro models as well. And so that's always something that needs to be weighed up. But new methods are coming through to address some of these shortfalls. In fact, very recently, test guideline 496 which is known as the irritation kit using an artificial membrane barrier, has been released, and that's going to significantly help in terms of some of the drawbacks around subclassification. Now, skin and eye irritation and skin corrosion are fairly straightforward processes in terms of their biology. They cause simple damage to the cells, relatively speaking, and it tends to be a reversible process. So once the, corrosion isn't, but irritation is a reversible process. And so once recovery has occurred, that there wouldn't be any long-term ramifications of that. Whereas for skin sensitization, that's a much more complex biological pathway. And also, once somebody is sensitized to a cosmetic ingredient, they will have that condition and an allergy to that chemical for life. So it's really important that we have an appropriate assessment for skin sensitization in place. And this slide just shows a schematic diagram of the skin sensitization pathway. And it's comprised of a number of steps in the skin. The first step is that an ingredient will come into contact with skin proteins and it will bind to those proteins. And then that in turn causes an activation of our skin cells underneath the skin barrier known as the keratinocytes. And then we get a further crosstalk between the cells in the skin, and our immune cells, the dendritic cells, then become activated. They will then migrate to our lymph node, and then we will get proliferation of lymphocytes, which is the basis of an allergic reaction in the future. So we have a number of key events in that sensitization pathway. And over the last decade or so, down to a truly global effort between a number of labs in different countries, we now have tests available to mirror many of those key events. And I've listed on the right-hand side here, you can see that there are key tests available for key events 1, 2, and 3. Event 5 is the one that was measured by the previous animal study, and I'll perhaps say a bit more about that shortly. But you can see that these tests were developed as a truly international cooperation, with labs in Switzerland, the US, and Japan all playing a role. Current regulatory guidance favours the 2 out of 3 approach for skin sensitization testing. And it may be an obvious question, why do we need to do more than one test? Why do we need the 2 out of 3 classification? Well, as I just mentioned, skin sensitization is a very complex biological pathway. And when we're replacing an animal test with tests that are taking place in vitro in the laboratory, we need to make sure that we are modelling all of the relevant aspects of that pathway. And so by putting together the results of these 3 tests, we're able to get a clearer picture of skin sensitization in the human than we ever were by doing one test in the animal, because we're able to look at the mechanisms of what's going on and the different points in the pathway that are affected by different cosmetic ingredients, which is really helping us to raise our collective understanding of how skin sensitization works. The tests I talked about just now are the ones that have full regulatory approval already. There are other tests coming through the pipeline, and a really interesting one is the GUARD skin assay. This test is a genomic screen, which is using a really exciting technology to look at more than 200 different markers of sensitization in human cells. And we're looking at the immune reaction in the skin with this test. So it's equivalent to the other test called the HCLAP, which models key event 3. So if we just kind of bring all this together and look at some skin sensitization results for a cosmetic ingredient, the picture on the right-hand side at the top shows some human skin cells being cultured in an environment completely free of any animal-derived ingredients. And the graph shows that if we incubate these cells with increasing concentrations of a cosmetic ingredient, we have an increasing amount of induction, so we're switching on a specialized gene which has been inserted into these cells as a marker of any chemicals that cause sensitization. So here we're getting a clear dose response. So as we increase the concentration of the ingredient, we get more of a response. And if we compare and combine those results of the keratinosense test for skin cell activation and combine those with the DPRA test for binding to skin proteins, we can see that the DPRA also gave us a sensitizer classification because it gave a level of as binding to the skin proteins that would classify that as a sensitizer. So here we have two tests which have both given us a positive classification as a skin sensitizer, and so we're able to conclude in our 2 out of 3 classification that this is a sensitizer. If we had had one result that was positive and one that was negative, we would need to go on to do the third test in order to get a conclusive result. But doing 2 tests first is very common because it helps to reduce the cost of the testing, although some companies do choose to do all 3 to get more complete information. So I just wanted to give you an overview of some of the tests that are being used. I hope that was useful just to get a flavour. I wanted to go on now to think about how the tests are being used to guide safety assessment. This is kind of the so what, really, because it's one thing to go ahead and do an animal-free test, but just like the traditional animal test, the important part is what do we do with the result? How do we interpret that? And how do we put it all together as part of a safety assessment? So in terms of guidance on when to use the tests and which ones are appropriate, there is guidance available, again, from the OECD, and these have been international efforts, again, over the years to develop what are known as IATAs, which are integrated approaches to testing and assessment. And there are some key ones, which again, I've added links here for anyone who's interested to check out later. But some of the ones that are appropriate for our discussion today are for the eye damage and irritation, skin corrosion and irritation, and skin sensitization. We'll look at an example in a moment. Just important to recognize that every ingredient of a cosmetic should be assessed, and then that forms part of the overall safety assessment of the finished product at the end of that process. So in terms of the IATAs that I just mentioned, without going into detail in this context today, this is just an example on the right-hand side of the kind of flowcharts that are available in the guidance documents to help any testing laboratories, any companies who are developing cosmetic ingredients, and their safety assessors to understand when it's appropriate to use the different types of tests. And in general, these approaches have 3 parts to them. Part 1 is a review of the existing data. So we may be using something for which there's already a lot of historical information available. There may have been previous testing. That could include historical animal data that with testing that took place before any relevant bans came in. But also looking at literature, looking at previous in vitro tests, and also looking at in silico information that has been generated through computer modelling. Now that is also used to pull everything together and to look at a weight of evidence analysis in part 2. So we look at all of the information available and then try to establish what additional testing is required. Because for many ingredients, actually, there isn't any additional testing required, and so that simplifies the assessment process. But if there is additional testing, these schematic diagrams and the accompanying descriptions in the IATA documents go into detail of how to decide which tests are appropriate and which order they need to be conducted in. So that's all very well for tests that we already have a well-defined structure, a well-defined process, and tests available to meet those endpoints. But what happens when there isn't a suitable regulatory test available? And how do we get around that when we have a ban on animal testing? Well, one of the key parts to solving this puzzle is really to think about using non-regulatory tests to build a weight of evidence about the safety of the product or ingredient. Quite often regulatory tests may not be an option, either because there isn't a test available. So, for example, for endpoints like the repeat dose and chronic exposure to ingredients, for reproductive and developmental toxicity and carcinogenicity, there are currently no fully approved non-animal tests available. These tests don't tend to be a requirement for the majority of cosmetic ingredients in Europe, because EU REACH only requires the test that I discussed previously for anything that is imported into the EU in quantities of less than 10 tonnes. But there are further annexes to REACH that describe what tests are required if a lot more of that chemical is being imported on an annual basis. And the premise of that is that the overall exposure in the population will be higher, and therefore there's a greater information requirement. Also, sometimes we find that the ingredient or product might not be compatible with the test that we've got. For example, they might not dissolve in a particular solution due to their chemistry. So it's really important that we have a whole toolbox, a whole portfolio of tests available. And the weight of evidence approaches are catered for in the majority of regulations around the world. They can't be used as standalone evidence if they're tests that have not yet gone through an international validation programme. But there are a number of stepping stones towards international validation, and there are a lot of very innovative tests with strong scientific credibility and reproducibility, and those tests are able to be used not as a standalone, but to support a whole weight of evidence around the safety of the product. It's also possible to use historical data, as I mentioned before, and also it's very common now to use read-across approaches from similar chemical structures. And to support this, in silico approaches, so computer simulated models, have really developed in recent years. You may have noticed in the earlier slide where I was listing the various human health endpoints under Annex 7 of REACH that I had listed acute toxicity, and yet there were no tests available when we looked at the OECD test guidelines. And I wanted to come back to that now, just to show an example in terms of weight of evidence approaches, because acute toxicity is the only key human health endpoint in EU REACH Annex 7, for which there is currently no regulatory animal-free test. Cosmetic ingredients under the test ban in Europe have to be assessed using a weight of evidence approach. And this can sometimes involve the read-across that I mentioned earlier. But we have developed in our lab a non-regulatory screen, which some of our companies that we work with are using to build a weight of evidence. It's using human cells in animal product-free culture and has been validated in our lab for 20 commonly used cosmetic ingredients. Now, this is only a starting point, but it's sufficient to be used as part of a weight of evidence approach. What we're doing is taking the in vivo LD50 value from the notorious LD50 test that you've probably all heard of. These were developed for acute toxicity using predominantly the rat, and that test uses a procedure to define the lethal dose for 50% of the animals. That has been widely criticised in terms of its scientific relevance to humans. What we're doing here is to try to create a more human relevant model by looking at the exposure of ingredients exposure of human cells to these ingredients, and looking at the validation we've done to make a prediction of the class for oral acute toxicity for that ingredient. We recognise that this is only a very early step. I mentioned right back at the beginning that using single layers of cells in culture isn't really a valid alternative to using a full intact organism. What we need to do is to build on this test, and we need to be able to predict more accurately by doing a more thorough validation of different types of chemicals. And we also need ultimately to build this in to a complete testing strategy, or IATA, for oral acute toxicity. So it's a starting point, and it's available for weight of evidence. Just to sum up, we're always very interested in looking at the scientific credibility of the non-animal tests and how that stacks up against the traditional animal tests. And in fact, any of the test guidelines that I've mentioned throughout this presentation have been through international validation trials where they've been validated against the traditional animal test as a benchmark. There are some flaws in using that approach because we know that the animal test models themselves are flawed. And those animal test models over the decades were never validated in the same way as today, where we have really stringent measures in place to validate new tests coming through. So here we have a situation where we have animal tests that have become the benchmark, but those benchmarks were never validated, and they have their drawbacks and their flaws. So now we have the new advanced in vitro tests, which are looking at human physiology, but we have to validate them against something, and we can't validate them against human data because we simply can't do those human studies, for these type of chemicals, and it's very, very difficult to get hold of reliable human data. So we're forced to use the animal data, historical animal data, as a benchmark. However, I do think that this has some use in the sense that industry has a lot of familiarity. They're in their comfort zone to some extent with these tests that have been used for so long. So if we can demonstrate that the animal-free methods are at least comparable, then that helps to build confidence in their performance. And just as an example, I've taken here the 3 non-animal tests for skin corrosion that I mentioned in a previous slide. And we can see how their performance stacks up against the Draize test, which is the animal test for skin corrosion using the rabbit. Just for those not familiar, sensitivity values look at how good the test is at predicting a positive result. Specificity looks at how good the test is at predicting a negative result. And then accuracy is a combination of the two. So it's an overall measure of the performance of the test. And you can see that these are strong. There's a strong correlation. That the best correlation is seen in this case using the reconstructed human skin that I showed you. But what do we do when the result of the in vitro test doesn't match the result of the historical animal test? Which one do we trust? And I think that this has been a question that's been increasingly debated. And for that reason, there's a strong move in the industry now to move away from simply the concept of having to replace the animal test. If what we're doing is just trying to find a replacement for a test that we recognise as having its flaws, we're not really letting go of that and moving forward into more human relevant approaches. So there's a definite trend to move away from trying to benchmark everything against the animal tests and to really adopt these new technologies at a whole new level to look at human relevance. And in that context, there's a trend towards exposure-led safety assessment. In other words, we might have a positive or negative result for all of the endpoints that we've discussed today. What does that mean in terms of overall exposure in the human body? What happens when that ingredient is absorbed across the skin? Does it go into the bloodstream? Is it excreted by the kidneys? Is it metabolized in the skin or in the liver? What happens when a fragrance ingredient is inhaled? And now what's happening is that the field is really moving beyond the simple hazard identification, which is the main part of what we're talking about today. But certainly some of the leading companies internationally are now collaborating to really look in great detail to generate a lot of data using lots of non-regulatory weight of evidence type tests, and to understand the disposition of ingredients if they are absorbed into the body. So just to sum up, I just wanted to look at some key points on why we would choose to use animal-free testing. As we all know, there are ethical drawbacks. There are a lot of animal welfare concerns around animal cruelty that's been going on for a long time now, and consumer demand is only increasing around the world in this area, and in fact has led to a number of bans on animal testing for cosmetics in many countries. And also, I think it's even more evident this year with the COVID-19 issue that we all have, is that it's very evident that there is a global trend towards ethically driven businesses and thinking about how we're all doing business, how does it have an impact on society and on the environment. There are clear scientific advantages in using non-animal methods, and I hope that I've demonstrated that to you by giving you a flavour of the type of models that are used. They're a much better model of human physiology. They provide detailed information about specific mechanisms of action. So we're able to see much more clearly, for example, when we're using human cells, what's happening in those cells. And we can see much more clearly compared with using a full animal model. And we can understand toxicity on a cellular and molecular level. And this all helps us to make more accurate predictions of the safety profile of products and ingredients for humans. There are also commercial advantages in using these methods. Many of the methods take only days and some take weeks versus months for a typical animal test, with a commercial result that we reduce the time to product launch. Using these tests enables access to markets around the world that have bans in place, including the European Union. The costs can be much lower than some of the lengthy animal tests that are required. I've written here can be, because it isn't always the case and it actually varies by the test. So that's something to consider individually. And then the EU cosmetic market, I think it's important to note that it really is thriving 7 years on from when the ban was fully implemented in 2013, showing that any of the challenges have been overcome and that more innovation has been stimulated through the ban coming into place. And then finally, we have some environmental observations. And actually, conducting animal-free testing has a much lower environmental impact than using animal test facilities, which use a lot of energy, chemicals, and waste. So just in conclusion, we have a wide variety of animal-free tests that are available using advanced scientific technologies. These have increased relevance to human physiology compared with the traditional animal tests. And I hope you'll agree that alternatives are now becoming the mainstream at an international level. So perhaps the word alternative doesn't do them justice anymore. The international regulatory approval has been achieved through the OECD Test Guidelines Programme, with a lot of global harmonisation in place already. And there's guidance available also through the OECD on how to apply the tests and when they're appropriate. There's a trend away from the simple replacement of flawed animal benchmarks towards more complex assessments of human exposure. And that's really a different topic that perhaps we can explore another time. And overall, animal-free testing presents a large number of ethical, scientific, commercial, and environmental advantages for companies and countries that are adopting their use. So just to close, I'd just like to go full circle and go back to think about why are we doing all this? And our goal is to improve human safety without making compromises on animal welfare or on the environment. So thank you very much for listening. If you wanted to pick up on any of these points with me afterwards, please do feel free to get in touch with me. I'm always happy to discuss more. Thank you.
Thank you so much, Carol. I think I'm switching over to my screen now. And if everybody can hang on for about 10, 15 more minutes, I'm going to take you through a quick overview of the regulatory trends around the world and some of the legislation taking place in the US. And I will just kind of start from here. I think my screen should be showing now. If not, somebody put something in the questions. There is still the question section on the side. If you have questions, go ahead and type those in, and we will have time to get to those at the end. Just real briefly, for those that you don't know, Cruelty Free International is an animal protection organization that focuses solely on ending animal testing worldwide. And we are also the oldest, originally established in the United Kingdom in 1898, so almost 120 years ago. We were originally known as the British Union for the Abolition of Vivisection, and in 2015 we rebranded to reflect our international work, and we now work with governments and regulators and companies all over the world and as part of international coalitions under the name of Cruelty Free International. We also founded Cruelty Free Europe, which is a Brussels-based network of animal protection groups working to bring animal testing to an end across Europe. And there we go. Also, just real quickly, we do have this resource available. It goes over some of the alternatives or non-animal tests that Dr. Treasure did in her presentation. It's a guide that I can send electronically, and I also have a few hard copies that goes over some of those tests and compares them to the animal tests and some of the information. So that's available if you want to. A copy of that, I can send it to you. And as Dr. Treasure mentioned, while interest in the development of non-animal testing methods really began in the 1980s, it was the regulatory changes in the European Union in the early 2000s that really was responsible for the explosive growth in alternatives. And the European Union's ban kind of came in a phased-in approach over the course of several years, starting in 2004, which saw a ban on the testing of finished cosmetics products. And then between 2004 and 2009, there was sort of a rolling ban and ending animal tests as soon as alternatives were validated. In 2009, saw a ban on the testing of cosmetics, a total ban on testing cosmetics ingredients on animals throughout the EU. And in 2013 is sort of was the final phase, which was the ban on the imports or the marketing ban, so that cosmetics with newly tested animal ingredients can no longer be sold in the European Union. And another keystone was in 2016 was sort of an upholding the integrity of the European Union ban when the Court of Justice confirmed that cosmetics that contained ingredients that were tested on animals outside of Europe could not be sold in the EU. So you couldn't take an animal testing data and use it to substantiate the safety of a ingredient in the EU just because it took place outside, the test actually took place outside the EU. So after that, the European Union really set off a trend around the world with other countries moving to match the progress on this issue. This map kind of shows the state of play in a color code. You see the hot pink color here shows the most complete bans that are in place. It reflects the prohibitions in place on the testing of cosmetics products, so the actual act of conducting an animal test is prohibited, as well as a prohibition on the sale of a cosmetic product containing ingredients that contain newly tested animal products or animal testing data for the purpose of complying with cosmetics regulations. So a marketing and sales ban, and that includes the United Kingdom, the European Union, India, Israel, Norway, Turkey, Switzerland, Serbia, and then we have a little blowout. We'll get into the states later. It shows several states in the US, California, Nevada, and Illinois fall into that category. The next color is the orange, and those are prohibitions in place on the testing of cosmetic products. So it doesn't have those are a ban that doesn't have the sales ban in place, and that includes New Zealand and Guatemala, and then several states within Brazil have that level of ban, as well as some states in the US which have what we call mandated alternative laws, which is New York, New Jersey, Virginia, and California already had this law in place. And that law says that if an alternative exists, the animal test can't be used, and that those laws actually extend beyond cosmetics in those countries. The next color on here, and states, shows turquoise, and that is a prohibition on the use of newly derived animal testing data for ingredients in cosmetics, and it only covers ingredients that are solely used in cosmetics. So if the ingredient's used in another product, it doesn't cover that, and that's Australia. The light pink shows where there's some legislation under consideration, and or has been for several years: Canada, US, Mexico, Colombia, Brazil, as well as in the states of Hawaii, Maryland, and Virginia. And we'll go to state action next. We began working at the state level in 2014, partly because of slow movement at the federal level. And that really inspired individual states to take action to introduce legislation aimed at phasing out the sale of animal tested cosmetics within the states. And this has really paid off. On January 1st of 2020, cruelty-free cosmetics laws came into effect in California, Nevada, and Illinois. The new laws prohibit the sale of any cosmetic product that's been tested on animals after January 1st, 2020. Other states, including New York, Hawaii, Maryland, and Virginia, have had cruelty-free cosmetics legislation introduced and debated. And then the states of Virginia, New York, and New Jersey, as I mentioned before, already have mandated alternative laws. So those are the laws that prohibit the use of an animal test if an alternative is available. And this has been really important because history has shown that state activity often leads to changes at the federal level. And indeed, these state laws have really served as a catalyst to bring stakeholders to the table. And many of the key provisions that were worked out in these state laws have now been adopted into the new Humane Cosmetics Act, which is the federal bill, which would end animal testing for cosmetics across the US. And that leads to the next slide. The new Humane Cosmetics Act, it was introduced in November 2019 in both the House and the Senate. In the Senate, it was introduced by Senator Martha McSally, along with Senator Cory Booker, Rob Portman, and Sheldon Whitehouse. And on the House side, it was introduced by Representative Donald Byer, along with Tony Cardenas, Paul Tanaka, and Ken Calvert, and that bill now has several co-sponsors. And this new bill, it represents a more polished version of bills that have bared the same name in the past. And these new versions it's informed by lessons learned from international regulations as well as the state actions. The language makes it clear that there would be a prohibition on conducting animal tests for cosmetics, so a testing prohibition, as well as selling cosmetics that rely on new animal testing data. So it has the sale prohibition as well as moving those products across state line. The bill does make some exceptions for certain, and I do have a more detailed frequently asked questions list for those that are with legislative offices. I'm going to just briefly go over some of the exemptions and other provisions of the bill, and I have more detailed frequently asked questions that goes into more detail on when these separate exemptions could be triggered. But just briefly, there are some exemptions for special safety concerns, and that would be in the rare case where there's a specific human safety concern that could not be resolved without resorting to an animal test, and that would be a test that would have to be approved by the Secretary of Health and Human Services. For foreign requirements, it would mean that the bill would not prohibit a company that tested its cosmetic products in order to sell in a foreign market where cosmetic animal testing might be required, but they couldn't turn around and use that new animal testing data in order to substantiate the safety of their product for sale in the US. So it wouldn't be able to be used as a loophole. There is a exemption for drugs. If a cosmetic contains a drug, the drug is regulated separately, so it's not considered a cosmetic. So if there are drugs included inside the cosmetic, then those are not subject to the animal testing ban. And then dual use ingredients. This sets out stipulations for what happens when an ingredient is used in a cosmetic, but then that same ingredient is also used in another non-cosmetic product where animal testing might be required, and under what circumstances would that data be able to be used. It sets forth some restrictions on that to ensure that cosmetics is not the cause of animal testing, and that using animal testing in other sectors isn't being used as a loophole to get around the cosmetics testing ban. And that leads right into other provisions. An important provision is some labeling provisions, which sets forth when a claim of cruelty-free or any kind of logos that claim that the product was not tested on animals. That's really in place to ensure that there's some consumer transparency around the use of any of the exemptions. There's also a requirement for the FDA to develop a strategic plan to promote the development and expedited review of new scientifically valid animal-free tests, and some reporting requirements to reports to the Senate and House committees and made publicly available about the strategic plan, how the act's being implemented, and information on whether any of those exemptions for safety or dual use have been used. There's also language about penalties to enforce the law. There's a civil penalty of not more than $10,000 for each violation. And of course, no progress would be possible without strong public support. And when it comes to banning animal testing for cosmetics, US voters are really united on this issue. A September 2019 poll conducted by Survey USA showed that nearly 8 out of 10 poll respondents said they would support a federal law that would prohibit animal testing for cosmetics. When broken down by self-reported party affiliation, we see that this is something that is shared across party lines and as well as across generations. This is something that even boomers and millennials agree on, high level of support across age groups and political affiliations. We see the same thing around the world. In May 2019, Cruelty Free International, along with The Body Shop, made history with the delivery of a petition containing over 625,000 signatures in support of a ban on cosmetic animal testing. And this was the largest petition delivered to Parliament in over 70 years. And then later that same year, we took 8.3 million signatures to the United Nations, and that was one of the largest petitions ever delivered to the UN. So this is really a global issue. It's a global industry, and consumers around the world are united on being interested in ending animal testing for cosmetics. I thank you for sitting through and paying attention to our presentation today. If you have questions, you can email us privately, as well as there's still a questions bar on the side. I'm going to turn my camera back on so I can talk to you in person. And there's Carol again. So I think we have at least one question on the side. Of course, remember you can email us privately if you want, and we have more detailed information. There is one question. It looks like it's for Carol. I think we have time to answer if you just want to read off the question, and for those that are still listening with us. Or is your microphone on, Carol?
Yeah, I'm just come back now.
Okay.
Yeah, it said, the question says, we often hear that we cannot replace animal testing until NAMs are in place, but would you agree that we need to think about that the other way round? For example, would you agree that the EU taking a stand to outlaw cosmetics animal testing, even where all the endpoints weren't yet covered by NAMs, drove investment in the development of NAMs? Yes, is the short answer to that. I do think definitely the consumer pressure has been at the root of all this, but then that leads to the regulatory change, and then that has forced industry to know that there's actually a deadline, and that money needs to go and be invested into the innovation so that we can meet the deadline. And I think that this is a lesson that we can take forward, where I was talking about the other tests that aren't yet available, because I believe that we do have the ability as a scientific community to solve those issues too. And so we just need to keep up that momentum. I definitely think that regulatory change is important, just as happened in the EU, to really focus industry's mind on progress.
Okay.
Anything else?
I think that's the only question that we have that's come through. Once again, thank you everyone for joining. It was really exciting to have such an international audience, and this recording will be available, especially to for your colleagues that maybe couldn't make it, that were interested. And thank you so much again, Dr. Treasure, for your great presentation. And we just hope that anybody who's still on, if you're part of a legislative staff, make sure you ask your boss to become a co-sponsor of the Humane Cosmetics Act. And we're really closer than ever now to achieving a cruelty-free world with so much industry support, innovative science, public support. We really are really close to finally putting an end to animal testing for cosmetics once and for all.
Thank you, Monica.
Thank you.